Morphological changes in apoptosis. In this video, we will begin by understanding the morphological features of apoptosis, the visible and molecular events that set it apart from necrosis or autophagy. We will look at cell shrinkage, the very first stage of apoptosis, where cells actively reduce their volume while keeping the plasma membrane intact. Next, we will explore chromatin condensation or pyknosis, where chromatin is compacted into dense heterochromatin along with the nuclear envelope. We will then move to the karyorrhexis, the fragmentation of nucleus into membrane-bound units, safely enclosing nuclear material. After this, we will examine membrane blebbing, dynamic surface protrusions that eventually give rise to apoptotic bodies. We will discuss how apoptotic bodies package nuclear fragments, cytoplasm, and organelles into sealed vesicles for safe disposal. And finally, we will see how dying cells are cleared in an immunologically silent manner through phosphatidylserine exposure and phagocytic recognition, preventing inflammation and maintaining tissue homeostasis. Research goal. At Research Goal, you will find research-driven courses that offer real research skills, data analysis training, and publication support. And best of all, it is incredibly affordable, costing less than other platforms. Join Research Goal today for free. Check out the link in the description box. Let's understand the morphological features of apoptosis. Apoptosis is the visible and molecular event that distinguish it from other forms of the cell death like necrosis or autophagy. These features not only serve as powerful diagnostic marker but also offer deep mechanistic insights into how cells commit to this tightly regulated self-destruct program. The first visible sign of apoptosis is cell shrinkage. In contrast to necrotic cells, which swell due to compromised membrane integrity, apoptotic cells, they actively reduce their volume. This shrinkage results from the loss of potassium chloride and water through ion channels, a process which is regulated by early apoptotic signals. Importantly, the plasma membrane remains intact, preventing the release of inflammatory mediators. So overall, what happens is, in case of apoptosis, cell is going to reduce its volume, and that happens because of the potassium ion, chloride ion, and also because of the loss of the water. And the important part also in case of apoptosis is the plasma membrane remains intact. And because of that, all the material remains inside those specific bodies, and that is not going to cause any kind of autoimmune response. If those components are released out, that can lead to autoimmune response or a potent inflammatory mediator release. Another important change is chromatin condensation, also known as pyknosis. During this process, chromatin is compacted into dense heterochromatin, which aligns along the inner nuclear envelope. The condensation is driven by activation of endonucleases and chromatin modifying enzymes. Under a microscope, it appears as deeply basophilic crescent-shaped regions, which is one of the most earliest nuclear changes seen in case of apoptosis. So that is another important morphological change that we see, where the euchromatin, which is the most loosely bound chromatin material inside the nucleus, it's going to get compacted into dense heterochromatin, and that is one of the most important visible change that you'll see in case of nucleus, and this is the indicator of the apoptosis process. As apoptosis progresses, the nucleus undergoes karyorrhexis, a nuclear fragmentation. The previous condensed chromatin breaks into several membrane-bound nuclear fragments. This fragmentation is managed by caspase-activated DNases, CADs, which cleave DNA and dismantle nuclear structures. This step ensures that nuclear material is safely enclosed, preventing exposure of DAMPs, which is damage-associated molecular patterns. Now, this is another fascinating change that you'll see in apoptosis, where all the material that is present inside the nucleus is going to get fragmented, but it's going to get, arranged into compact vesicles so that there is no release of damage-associated molecular patterns, and the immune system remains silent. Simultaneously, the plasma membrane begins to show blebbing. It's a dynamic bubble-like protrusion. This is driven by contraction of the actin-myosin cytoskeleton and regulated by caspases and Rho associated kinases. As apoptosis proceeds, these membrane blebs stabilize and contribute to the formation of apoptotic bodies, which package the cell into smaller digestible units. I think this is one of the key signature of apoptosis, where you'll see those smaller apoptotic bodies, and those are visible bodies. Inside those bodies, you will have all the material that is important for the cell, and that can cause immune reaction. So the cell is going to dissociate into smaller bodies, and those will have intact plasma membrane, so that the material is not going outside, material remains inside, and there is no immunological response that is present inside the body against those particular material, because the material is never going to go out, and immune system is not going to interact with that particular material ever. That is why the apoptosis is immunologically silent. These apoptotic bodies are membrane enclosed vesicles containing fragments of nucleus, cytoplasm, and even intact organelles. The formation marks the final morphological stage of cell disassembly. The formation of different compartments facilitates efficient clearing by phagocytosis and preventing the release of harmful intracellular content. A key reason that is why the apoptosis remains non-inflammatory. The next question comes, what will happen to those apoptotic bodies? Those apoptotic bodies will be phagocytized by the phagocytes, and the entire structure is going to be processed inside the phagocytes. So there is no allergic reaction, there is no inflammatory reaction in the body. So that is why apoptosis remains non-inflammatory. Perhaps one of the most elegant features of apoptosis is the immunologically silent clearance of dying cells. Apoptotic cells expose phosphatidylserine on the outer leaflet of their plasma membrane. Phagocytic receptors such as TIM4, BAI1, and stabilin-2 recognize these signals and mediate engulfment by macrophages or neighboring cells. This process avoids triggering inflammation, unlike necrosis. However, when apoptotic clearance is defective, apoptotic debris may accumulate. This can result in autoimmune response due to the immune system recognizing exposed self-antigens as foreign molecules. So because of the interaction between the surface receptors and the phagocytes, they are able to take those apoptotic bodies and then digest them without causing any activation of the immune system. But if that is defective, if the apoptosis is defective, what will happen? All the component is going to go out. It can interact with the various immune cells and causing allergic reaction, inflammatory reaction, and that is not good. It can lead to autoimmune diseases. So that is why apoptosis is one of the fundamental processes of our body, and if it is regulated, then that means our process, the immune system, is working fine. So our entire system has a complete and regulated process of taking care of the waste material, dead cells, without causing any kind of inflammatory response. Alright everyone, in this lecture we explored the key morphological features of apoptosis, starting with the cell shrinkage, followed by chromatin condensation and nuclear fragmentation. We then looked into how membrane blebbing leads to the formation of apoptotic bodies, and how these are cleared through phagocytosis in a silent, non-inflammatory manner. Together, these steps highlight how apoptosis is a highly regulated process that ensures safe and efficient removal of dying cells while preserving tissue homeostasis. So I hope now your understanding of apoptosis, especially the morphological changes that happens in the process of apoptosis, is clear. We will be talking in detail about the biochemical as well as molecular changes of apoptosis, and your understanding of apoptosis is going to get cleared. So stay tuned. Please watch all the videos. I'll be meeting you in the next one. Till then, take care everyone.